All right, so here we are in part 2 of this chapter 11, dealing with nucleic acid. So I want to just follow up with what we had just ended on the previous one, DNA and RNA. So here's a great example of a great pictorial, if you will, these cartoons of RNA, the ribonucleic acid, and DNA, deoxyribonucleic acid. And you have a single helix and you have the double helix, with most of the base pairs, having some kind of commonality there, with the exception of these nucleobases, uracil and thymine, which are unique. Uracil for RNA and thymine for DNA. To get more information on the importance of RNA, I recommend going to the RNA world because they're going to give a lot of interesting insight. In fact, there is some strong arguments that it wasn't DNA as being the origin molecule material, if you will, of life, but actually RNA. And how they support that this kind of theory, well, they use a bunch of different evidence that has been compiled from different areas, including from meteorites, but they also look at, for instance, different species of viruses, for instance, that may exist in certain lakes and whatnot. And so there's a lot of good information if you go to this RNA world, and even in a more recent publication here, and I just have this hyperlink as novel RNA genome. I think it'll be pretty interesting if you want to check that out. But because RNA is smaller, and because it was one of the original templating molecules that was used to be able to try to expand and reproduce and just grow, I guess, additional DNA structure to amplify it, they call it, in the laboratory in order to sequence it and then be able to make some kind of scientific assessment on it. This is some of the reason why this is even any, a concern at all. And of course, viruses, if they have, RNA, they're going to be considered retroviruses, and we'll talk about that in a moment. But those are the more important ones. And viruses are ubiquitous, and they've been around since the dawn of time, essentially. And so there's a lot of soundness to saying that RNA is probably the root of life, at least the predecessor to the DNA molecule, and the discussion therein about that. So let's talk about protein synthesis scheme, just from a general point of view. You're going to have a couple different processes going on. You're going to have, first, you have to start with DNA. You're going to have some transcription. We'll talk a little bit more about that in detail. That kind of activity that's going to go on, and that goes on within the cell, is going to produce this mRNA, which will be leaked out of the cell, and then it'll be consumed by the ribosome, which is going to travel along it. And there's a couple different units here, individual units that come together. It's almost like a sandwich, right? But it's like a machine that just processes and stamps stuff out. And depending on something else that's going to be tagged, coming in here, and it's going to be tRNA, you'll be able, which will be coded with a specific protein. You'll be actually able to sequence a series of these amino acids, which will eventually form a peptide large enough to call it a protein. So if we were to talk about this, we'd say, first of all, you need this transcription to occur then, because you're starting with the DNA, you have the double helix. How are you going to get that copy? How is that going to happen? Well, the DNA is going to be copied, just slightly though, using an RNA polymerase. So this is going to be an enzyme that's going to help create this mRNA molecule. All right, it's an enzyme that binds to the DNA, and it unwinds it just a little bit, maybe 4 turns, and it does so just enough to create a copy. That copy is called the mRNA. And there are some transcription factors that can be included along with that RNA polymerase to help make that go maybe more expeditiously or in a more targeted manner. That's fine. But the whole point is you get an mRNA copy that comes out, that is produced from the nucleus of the cell, right? And it travels from the nucleus to the ribosome, and ultimately proteins are going to be made. The translation then, and this is occurring within the ribosome, is going to have, it's going to convert a sequence of nucleotides into, a protein sequence in the ribosome. We'll talk about that, in just a moment. But it's important to know that this tRNA ribbon-like, they call it a linker or adapter molecule, comprises between 76 and 90 nucleotides. So it's larger than, for instance, the number of amino acids that would be found in, for instance, insulin. So this is still, it's still a large molecule. These tRNAs are still large molecules. The mRNA is still a large molecule. And, although they are smaller than the DNA, we're still talking about some stuff that, some molecules here that have some heft, okay? So let's talk about the 3 types of RNA found in cell transformation, right? Whenever you go from DNA to proteins, you have the, transcription, right? You have the mRNA is what we're talking about here. This is the gene copy that's made, and it has to be done with this RNA polymerase, has to be done. Cofactors can be involved, or I should say transcription factors, and you'll get that segment. The DNA template for transcription, if we can indicate this, then here's how it would go. You would have the DNA, and then you'd have this mRNA. The mRNA is going to be represented here in this blue bar, whereas the DNA is going to be this purplish bar. Remember that ribbon where we had the backbone ribbon of the phosphate and the sugars? Well, that's giving rise to that upper purplish kind of pinkish ribbon. And then you had each of those bases that were subtending from that, whether it was A, C, G, or T, right? Those are the 4 that are allowed for DNA. If we're talking about RNA, we still have those nucleotides, but then we also have the 4 bases. And instead of T, you have U, right? Uracil instead of thymidine. So those are the difference there, or thymine, I should say. So I'm thinking of I got amino acids on the brain. So once this has been completed, so you have the DNA, and again, this is in the nucleus. You have this mRNA, which is being developed right here, by virtue of the RNA polymerase and any other transcription cofactors that might be present. Yes, RNA polymerase is considered a transcription factors, but you can have other ones, and in reality there are. But once that is completed, once a mirror image, in essence, is being produced, then it's released into the rest of the cytoplasm, and the ribosome is definitely going to eventually find it and start to process it, provided the translation RNA, the tRNA, is present and is coded correctly. So the ribosome, as you know, we call it the protein factory, but there's actually an abundance of RNA plus protein, both in the small unit and the large subunit, that are responsible for this process. So it can't just be the proteins that make up this ribosome, it's actually part of the RNA. So if we're talking about the RNA world and everything else, and if I have to go back to that, I'm hoping that you're starting to see the importance of RNA and kind of, its positioning in terms of what is the importance of life, or what is the origin of life, I guess is probably better, not the importance, but the origin. And you would say, what is the importance of RNA versus DNA, or even just RNA in general in this world? Well, RNA is pretty important because there's a whole organelle that's based on a particular type of RNA, intermixed with some protein that might help it actually have some mobility through some intermolecular forces and stuff. So there's some interesting aspects there that we're not going to talk about here, but surely could be of interest. And so you have the ribosome, which takes that mRNA strand and it processes it. It processes it provided there's a tRNA molecule that is also present, okay? And so how do we talk about that? Here is how we talk about that. The tRNA, you're going to have this, remember, 76 to 90 nucleotide kind of sequence molecule, so it's fairly big. You would call this one inactive because it doesn't have a protein attached to it. This would be this would be a molecule that, for instance, is waiting to attach to some kind of amino acid. And it's going to form an ester bond, okay, with the amino acid. But whenever it hasn't formed that bond with the amino acid, then it's going to reside in its hydroxyl inactive state here. Okay, so that 3' end is going to still remain just, free, okay? Within this tRNA molecule, you're going to have this anticodon present, okay? And it's coded for 3 different bases there, and sometimes this is referred to as the anticodon loop. Some people refer to this as like a T-shaped structure, probably could. It could also be, I guess, kind of of a sideways R or even pistol grip, you know, kind of structure there as well. But the whole point is you have this tRNA, and oh, by the way, hydrogen bonds are giving rise to this kind of integrity of whatever that shape is, whether you want to call it like an R or a T shape or some kind of pistol shape, you know, but there's a lot of intermolecular forces going on. Hydrogen bonding is again reigning its prevalence within these biological molecules. So important it is. You have anticodons here, and we consider this to be a 3-base sequence. You can call it a triplet if you want to, with the anticodon loop, which I just mentioned, and it forms hydrogen bonds then with the complementary bases on the mRNA. This is important because, again, tRNA is completely separate than mRNA. They're going to come together. How are they going to come together? It's going to be the codon-anticodon relationship. Complementary bases are going to overlap, and it's going to be through hydrogen bonds. And when it's properly tagged, I guess that's easy to remember with transfer because it's also T there, then you're going to have an amino acid on one end, you know, forming an ester bond with this 3' free end. Guess what? It ain't free anymore, right? Once it's attached to a particular type of amino acid, not just any amino acid, but these tRNA sequences are programmed for certain types of amino acids. I think that's important to note here. Only when that is tagged properly, then this tagged RNA molecule is going to be admitted into the ribosome, where by virtue of the complementary base pairs, you know, this processing can occur, and then it's actually going to release then that amino acid from this tRNA, and the tRNA goes back and gets another one, okay? So each of the 20 amino acids has one or more of this tagged or transfer RNA available to bring them into the ribosome. It's almost like a ticket. You can't get to the ribosome unless you got your ticket, okay? Well, your ticket is a particular type of amino acids, okay? So that's pretty interesting that each of the 20 acids are going to have a unique sequence that's going to be dedicated to them. And that is, again, the only way to incorporate into the ribosome. And then the ribosome is going to be able to attach these individual amino acids and have them, facilitate that peptide formation between them. Obviously, if you get enough of them, polypeptide, and if it's so big, then you're going to get proteins going on. So let's look at this in detail. Here's another cartoon, and hopefully this makes even more sense. So I know we're talking about this ad nauseum and at length, whatever, but it's important that we understand what is going on inside this nucleus versus in the cytoplasm and then at the ribosome. So inside the nucleus, again, we have messenger RNA. It's going to come in, right? Copies DNA strand via the RNA polymerase. That's one of the transcription factors. There could be others present to help expedite this process. And what does that mean? That means several turns of the DNA strand is going to unwind. Maybe about 4 strands going to unwind. It's going to be copied, then it's going to be shot out of the nucleus where it's going to float around in the cytoplasm until the ribosome finds it, okay? And then whenever the ribosome, attaches onto it, it's continuing to move. It's almost like it's an automatic stamping machine, right? It just continues to move along that mRNA. Nothing is going to happen though of any kind of substance, like any kind of substantive kind of benefit in terms of protein formation, unless those transfer RNA molecules, of which there's at least 20 of them, for each of the different 20 amino acids are tagged with the appropriate amino acid, at which point it has then received its tag for admittance, its ticket for admission into the ribosome. It's then able to be admitted. The complementary base pairs are matched up, held together through the hydrogen bonding forces, and the ribosome just continues to move along that mRNA until it can, these individual amino acids are then linked up, peptide bonds formed, and what happens to the rest of the mRNA? Well, it can get scavenged up and reused because that's valuable molecular, moieties that could be used for fuel sources, right? You have sugars, you have phosphates, and you also have nitrogenous bases. So you have all of that. Now, what happens to the tRNA, the transfer RNA? Well, that doesn't get gobbled up. That gets recycled. So it loses its protein and then immediately goes back into the cytoplasm, into the bulk of it, looking for more amino acids that it can pair up with. So that is how the protein sequences are then formed. So I think that's pretty interesting. So Again, to drive this home, I think, because I think that this is really important, it's easy to get lost in some of these details with all the different RNAs that there are. Again, you have a transcription. Number one, RNA polymerase binds to DNA and links nucleotides to construct mRNA. That's the first part. Second part is tRNA activation. This means that an amino acid is tagged to a certain nucleotide sequence between 76 and 90, okay? And there's lots of different ones. How many amino acids? There's at least 20 that we have in our bodies. Sometimes it's arguably that there could be up to maybe 22, you know, depending on if there's different kind of species. And of course, we're not talking about engineering of anything, like bioengineering of things, because that can introduce yet other factors that we haven't discussed. But then it goes, once it's been tagged, then it can enter into the ribosome because it has its ticket for admission, and then proteins can start to form. Now, here's where it's kind of interesting as well. So remember that the complementary base pairs still hold, right? Adenine, uracil, guanine, and cytosine, right? A, U, G, and C. And again, it would be thymine, right? T for DNA, but for RNA, it's U, uracil, okay? So that's the complementary base pairs here. Interestingly, the start codon on mRNA is going to be always AUG, and this means that the tRNA anticodon, that triplet sequence, must be UAC, if it's AUG, plus an amino acid. Which one? Methionine. It's nonpolar. It's essential to the human diet, but by the way, we don't make it. We have to get that through eating, for instance, like eggs. But here would be the structure of that. But that is one that we need in order to have proteins start to be formed. So it's interesting. It's like, which came first, the chicken or the egg? Well, you can't have translation unless you have methionine present. So you need to get that in your diet. So you can't form a lot of protein without having methionine present in your body, present in the cytoplasm, right? Where this anticodon is tagged with it, right? Where, I mean, not the anticodon, but the tRNA. It has to be tagged properly with the corresponding mRNA, you know, base pair matchup, and of course the ribosome is just there gobbling up and doing what it does. So you have the next codon on the mRNA along this matching tRNA anticodon fall in line next to the first, and then adjacent amino acids form the peptides and so on, and it just gets bigger. Again, how are they connected? Hydrogen bonding inside that ribosome, right? After the amino acids are linked together via peptide bonds, the tRNA is discharged from the ribosome to look for, again, its dedicated proper amino acid. Again, so you have at least 20 of these different tRNA molecules searching for their amino acids. And then the mRNA, well, it can be scavenged. There's other molecules that exist in our bodies that exist even in the cells that are used to scavenge that residual mRNA. And you know, what do we call that kind of motion whenever the mRNA is advanced in the ribosome? Or you know, you could even say the ribosome is advancing along the mRNA. They call that translocation. And then finally, termination, because eventually things, if they're going to begin, they got to stop, right? You have the ribosome encountering a stop codon, which is either going to be a UAA, a UAG, or a UGA, and it's going to be present on the mRNA. Guys, this is really interesting. We're going to talk about this in terms of retrovirals in a moment, but this indicates that there is going to be a stop sequence that is going to be present. And what that means is that protein synthesis can stop, okay? So methionine is excised from the polypeptide chain, then the polypeptide folds in tertiary structure, and you get going on with the normal types of proteins, and that gets distributed wherever it's going to be distributed to. Remember, we have different cells all throughout our bodies, all for different purposes, and you know, those polypeptides that do end up forming the proteins have their function too. So really interesting stuff, and it can get quite heady, but there's been a lot of work on here. It's absolutely amazing how proteins are created from the RNA and from the DNA processes. So again, you know, I had highlighted in green and in red the signals that start and ones that stop. These are just some codes, you know, and again, we're talking about RNA, so you're going to be using uracil, not thymine, okay? And then you have all these, you know, letters that indicate that, right? You have the nucleotides that are present there, so that's it. If it's going to be codons, and these are all triplets, okay, then you're always going to have a UUU or a GGG going to, you know, and corresponding to a particular amino acid. So in this table here, 11.3, whenever you have these brackets, like in the upper left on the UMP nucleotide, where you have UUU and UUC, and you see that they're bracketed and it has a PHE, well, that's considering the phenylalanine, right? Amino acid, or the leucine amino acid. That's what that is corresponding to. And these are the sequences that we now know. You can either use, 1 letters for the amino acids, as you know, or you can use 3 letters. So, but, it is now known that specific triplet codes do code for specific amino acids. Okay, so if you had this amino acid sequence, right? So you had a 5' and you have a 3', right? You have your start and you have your finish, but they're both free. You would say CAA. So you would come over here, look at table 11.3, look at the C, which is in the second row here of this CMP nucleotide, right? You just look over, you try to figure out where the CAA is. Oh, there it is, it's GLN. That is also denoted by the letter Q, right? Glutamine, right? So, and then you just have the isoleucine, and then you have the valine, and you can do all that. So, but that would be the end coding that you would be able to write in terms of what the sequence of the amino acids be for. This is some of the research then that Sanger won his Nobel Prize in, to try to figure out, well, what amino acids, what is the sequence of all of these different RNA combinations that are giving rise to these amino acids. His research has led to that. So the thing that is hot now is CRISPR technology, and CRISPR technology is really interesting because it basically, if I had to summarize this very shortly, and I'm probably not doing it good enough justice, but what it does is it takes bacteria's natural ability to excise and insert DNA, and it's our ability then to mimic that in the laboratory to be able to bioengineer, a given entity, right? A given, DNA kind of combination that allows for improved benefits. For instance, like if there's certain coding that was there that was indicating that there might be something cancerous or something, or some other kind of ailment that was undesirable that could be excised. You could implant some other coding, genetic coding, basically, to say, look, this could be normal, and then maybe that health infirmity would not have materialized. So obviously this is in a go back to starting zero, right? Starting with somatic cells, from like day 0 or day 1, it can't be something that we can do as easily with existing bodies without certain ramifications for this, but it is, a possibility, and I'm sure things are going to continue to evolve here. So, I have some links here. You can always check them out for the RNA and CRISPR. But this CRISPR here, it excises DNA at targeted places and relies on cell repair mechanisms, again, inspired from bacteria, to insert DNA sequence, but it's often not very precise, and so off-target, I don't want to say, mutations, but effects can occur, okay? And on the other hand, individual DNA bases like the adenine, guanine, cytosine, and thymine can be edited with deaminase enzymes. So you can do this with RNA, you can do this with DNA, But again, even these DNA additions or edits with these deaminase enzymes can also have off-target DNA consequences, So if you could do it with RNA and DNA and there could be off-target consequences, some of these might be okay, but some of them might not be. So this is still very much in the early stage of research, and a tremendous amount of funding has been going on with this, but this is the basis for genetic engineering now. And I have my Sasquatch here, I guess, my Bigfoot, right? My genetic mutations. This leads us to this, because we're talking about mutations, in terms of like whether it's an off-target, kind of effect where it was done in a laboratory, that's one way. But what about just natural mutations? Well, guys, they occur all the time. In fact, this means, from a DNA perspective, it's a change in nucleotide sequence that alters protein synthesis, okay? And most mutations don't involve protein encoding genes. So one of 3 things can happen. Either you have no change in protein sequence. This is referred to as a silent mutation, meaning that if you have UUU going to UUC, it's still the same amino acid, phenylalanine, didn't matter. So you can go back to that other table that I showed you a couple slides back on 11.3, and you can see that there's going to be some multiple options that of these triplets that can still give you an amino acid sequence, right? But it won't matter. That's called a silent mutation, because even if it is a mutation, it doesn't negatively affect us. Here's a number 2 possibility. The change in protein sequence, but negligible effect on protein function, okay? So this is also called a silent mutation, meaning that if amino acids happen to be exchanged, provided they bear similar characteristics such as polarity or charge, such substitutions aren't likely to affect protein function. So if AUU, for instance, isoleucine, is mutated to GUU, valine, it's because both are non-polar. Or again, we're going to call this a silent mutation because it doesn't do anything noticeable in our daily life or our operation, whatever, that would show that protein function, I should say, protein function has been malaffected. Or number 3, the change in protein sequence, but marked effect on protein function occurs. In this particular case, this is where, for instance, amino acids are exchanged, but they're different characteristics that affect protein function. For instance, something that is polar neutral or non-polar, or has a polar positively charged. I mean, all of that stuff can start making a difference, okay? This is important. Where do these mutation sources come from? Replication errors, okay? So this is just spontaneous mutation that can happen that could be initiated by the environment, whether it's mutagens or carcinogens. If something is too toxic and that's going to give rise to alterations of a given protein or a nucleic acid arrangement, or how that's, what's going on in the cells. Basically, if the cell can be affected and a protein can be degraded, well, then you're going to have mutations, right? And that can happen anyway. Viruses can also do this, okay? But RNA mutations, I mean, we're talking about DNA in this particular case, okay, as being, the focus here. I think your book even does that, but it's even more common to have RNA mutations. They're smaller, they've been around for a long time, especially if you subscribe to the RNA world of evolution kind of theory, where that preceded DNA formation, which kind of makes sense. I mean, RNA kind of, as you know, the whole transcription process, right? I mean, that's the first stage. You got to unwind a little bit of that DNA, make a copy of it. So RNA is already ideally suited to make copies of something that is much bigger than itself. And viruses are pretty omnipresent, that it would make sense that there would also be, some mutation potential there too. So with respect to genetic mutations, so you have the DNA, and again, this one's really just talking about DNA, but can also be RNA as well. In this particular case, your book is focusing on DNA, and that's fine. The DNA gets altered. If the DNA gets altered, then you're going to have, well, all the altered proteins, okay? If that will be considered a genetic disease, if it happens in the sexual reproduction cells, i.e. the germ cells, or it will be in the somatic cells, and this would be known as cancer, for instance. So this would be something that would have evolved over the course of time, all right? Something going on in our bodies, but maybe didn't initially happen whenever we were at age, 0.5 years, or 6 months old, or whatever it is. This has developed over the course of our lifetime, by virtue of various, reasons, the ones that I had just, mentioned. Here are some different examples of the genetic disease, the protein or chromosomal defect, and the disease symptoms. I will let you consider this on your own. This is in your book, table 11.4, but they give you a bunch of different diseases that are related to genetic mutations, okay? Deficiencies in the protein for whatever reason, okay? Whether these were, whether these have origin from the germ cells or whether they are somatic. Okay, so this is all part of it. Now let's talk about viruses, because this is really timely given the whole pandemic, going on right now with the coronavirus. Let's talk a little bit about the viruses. So, let's not forget, the purpose of a virus is to hijack the host cell for benefit of virus, okay, of itself, and you could consider it to be a parasite. Now, although we say that viruses are bad, right? In fact, the roots of the word, it has everything to do with toxic, you know, we ourselves are walking around, even if we're quote unquote healthy, we are walking around with fragments of viruses, just part of our DNA. Remember, our genetic makeup, only what, about 5% of our genes are coding for proteins anyway, compared to bacteria is like 95% of the genes are coding for protein. We are comprised of many different things, and some of those things are viral components. And some of those things are then not, giving rise to us living in a secluded lifestyle, with, artificial support trying to help us live a life, right? So although viruses, now, of course, I mean, nobody wants to have a virus. The viruses definitely are, can be and are toxic, to our livelihood. They have been around forever. And there's a whole bunch of these viruses that exist in the world. So we can't get too, I guess, overexcited about the fact that, well, viruses, are just bad. We should stay away from viruses no matter what and have hand sanitizer around us all the time. Well, I mean, it's just not reasonable, okay? Whenever we ourselves are living with viral components, fragments, if you will, within our bodies, it's just part of our, existence here in this world. We weren't the first ones here, and we hope that we're certainly not going to be the last ones. So anyway, so a little bit about the viruses. They have between 3 and 200 genes, okay? So smaller than the bacteria. Remember, the bacteria had about 2,000. And they are minimally designed. Really, they are, from an efficiency point of view, from an economic point of view, I mean, the juice is worth the squeeze when it comes to the design of viruses, especially if they're intelligent, because they don't have much, but yet they evoke such a powerful response because they make use of the host to be able to continue and propagate, right? So in this particular case, you would have this image, and we just have here this viral image. It has these protein spikes here. They can be different, right? There's a bunch of different structures on them. Usually they have about 2, maybe 3, maybe even 4 different types of protein arrangements, orientations. Could be more or less depending on the virus, of course, and I'm not a virologist, but you could definitely look into the research on it. But each one of these different protein spikes will be that anchor point for being able to penetrate into the host cell. So that's really the purpose of the protein spikes. This capsid here is a protein, hard protein layer. Not all viruses have this capsid layer, okay? I think that's important to understand. Usually the envelope here is oftentimes just made up of the phospholipids, that fatty, squishy stuff, all right? And then, of course, you have the nucleic acid that is inside of the virus, or it may be organized as RNA. That's going to give rise to a specific type of virus called a retrovirus. But if it isn't, then you're going to have these nucleic acids. Okay, so if this virus exists outside the body, it's generally considered to be a virion, okay? V-I-R-I-O-N, I have up there. Now, when it does puncture the host cell membrane to be able to deliver its payload, which is nucleic acid, and more in fact, if it's even more order, the RNA, then you're going to have it turn into and be referred to as the virus, right? But it does so via these protein spikes. Again, not all viruses have the tough capsid shell, and the capsids are generally tougher to break than the non-capsid viruses. Guys, the coronaviruses have lipid-based envelopes. This is the softer stuff. These coronaviruses don't have this capsid type of environment, okay? And because of that, because they have this lipid-like soft squishy layer, if we could squish them between our fingers, we would be able to. But this is why they're highly susceptible to being disrupted with, for instance, alcohol, because meaning hand sanitizer and whatnot, because it interrupts the intermolecular forces. Of course, once a virus, infects the body, then it's a whole different way of trying to address how to, how to combat that virus, and we'll talk about that in a moment. But as it is, just laying there, sitting on the table before it gets inside your body, as it exists as a variant, it's very easy to destroy, which is probably even maddening, because it's like it'd be a lot easier just to carpet bomb, the whole world with a hand sanitizer, then we won't have to worry about that. In some respects, that, yeah, I guess that could be true, but obviously there's there's a lot wrong with that, not to mention the logistics and everything else. Plus, I don't know whether I want to be bathing in hand sanitizer. Anyway, who knows? Okay, so again, this is a cartoon here where we have a virus infecting a cell. Okay, but again, before it's there, it's called a variant, but then it punctures the cell by virtue of its proteins, right, and its enzymes. It delivers its payload, which could be nucleic acid or RNA, and it starts to, utilize the cell's function in order to be able to create its own kind of DNA. Okay, and then it propagates throughout the rest of the body. This means that, once it does penetrate the host cell, that means everything is under its power. So when, it's highly under control. So just some couple comments here. RNA is small. It's used as a DNA template. Viruses are among the most ubiquitous species on Earth. I mean, you have archaea, which might be even in more abundance, but then like viruses are like right there, right next to it. And some people argue, well, what is a virus? Is it an organism? Does it eat and breathe and whatever else? Well, some biologists really refer to this, or some experts, virologists, even consider that viruses are an organism at the edge of life. So I guess that's a safe way of saying maybe, maybe not, but it's not fully known. What are some virus examples? So here, and these are from your book, 11.5, some diseases caused by viral infection. You have the common cold. Notice what type of virus is it? It's a coronavirus. And there's more than 100 types here. You also have the flu, which is different from the coronavirus, and they call that the influenza virus, and that looks different. You have herpes. You have leukemia and AIDS. You have hepatitis, mumps, and Epstein-Barr. And these are amazing, right? And here you have the structure. Look at this hepatitis C structure. Almost looks like the coronavirus structure in terms of its protein spikes, right? Look at that. So I have here the coronavirus, and this is on the Nature website publishing page where I got this. But this is the coronavirus right here. That structure, and it kind of looks really similar to the hepatitis. Notice again how they're saying that types A through C are the most common with the hepatitis. You guys probably have been inoculated against that, using the vaccines and whatnot, maybe to help prevent some of that stuff. But these viruses have really similar structures, okay? Interestingly, at least as of 2002, viruses can be made artificially. This is true. The polio virus is an example. And again, it was published in 2002 in the Scientific Journal. So it can be done. It is now the year 2020. So we have at least 18 years of being able to manufacture artificial viruses. Here are some other examples. Bacteriophages, okay? This is really interesting because at the turn of the 20th century, while most of the Western world was using penicillin perhaps to counter any kind of bacterial infection, which would later give rise to amoxicillin, in particular in Russia, their preferred avenue of antibacterial treatment included the application of bacteriophages, i.e. a type of virus, where you actually have the DNA payload already loaded in there within this bacteriophage, right? And you have this, make it like a mechanical kind of structure, and you could talk about it. Again, these spikes, those would be protein spikes, right? You'd have all these different aspects of this bacteriophage, but it could, it could deliver its payload to be able to counter the bacteria operation. So here is an image 200,000 times, in scale. You have all these tiny little bacteriophages that are attacking a bacterial cell. So you have these bacteriophages on the outside, and they're able to attach themselves and then puncture to deliver its DNA into the cell in order to wipe it out, right, to inactivate it. Just to let you know, bacteriophages, just like viruses, are ubiquitous on Earth, and there's even more of them than bacteria. All right, retroviruses, this is really important. So, here you have viral RNA instead of nucleic acids that are randomly put together. You actually have the virus actually contains RNA that's able to be, produced into a DNA package, once it infects the host cells. And then with some other things, you have this integrase, which is another enzyme. So you have a couple enzymes that are being able to get in there into the cell, being able to create, extended structure there, and just go on, this is just part of it. This is the type that is, for it to survive, reverse transcription needs to happen, and complementary strand copies of its DNA is going to be produced. Integrase is then used to integrate it with the rest of the host DNA, so now it can travel, right? Now it can exist in other parts of the body. Now, I'm gonna, again, come back to it, to our genome and our genes and our makeup. Did you know our genome is comprised of endogenous, meaning generationally passed via the germline, via sexual reproduction, retrovirus fragments, okay? Our genome comprises between 5 and 8% of these fragments, of RNA fragments. We are part retrovirus, all right? Endogenous stuff, though, that is okay, as opposed to exogenous, which can give rise to different complications and ailments, and then we die, all right? But we have some endogenous RNA that has been around since time immemorial, since your first family line, how far ever back that goes, okay? You are manifesting parts of RNA, okay, part of a retrovirus in your genome. Pretty fascinating. So how do you battle it? Well, there's a lot of different ways, depending on what type might show and or prove out more effective in a clinical trial, or maybe just for you, because the way you're going to respond to a drug treatment or to an infection is going to be different from somebody else, okay? So here is an example of HIV-1, right, virus, okay? How do we do this? Well, you can either block it by entry inhibitors, that's one way. Another way is that it can be blocked by nucleoside analogs that will interrupt the transcription from actually being able to create DNA from it. Integrase can be interrupted, right? Because you have inhibitors. Now we go back to our enzyme chapter with proteins, and we talked about competitive inhibition of different agents. Well, this is another way. And then you have the host DNA. If it's able to make it through, for instance, if the entry inhibitor didn't work, if the nucleoside analog didn't work, if the integrase inhibitor didn't work, and the host DNA helix was, double helix was able to form, and it's comprised of viral, product, and now, there's viral mRNA that's going on because it's replicating and stuff. Well, what else can you do? Well, you can block it by protease inhibitor. So there's a lot of different ways. Each of these are going to work to a varying degree, and most of the time you might need a combination, because maybe one of these isn't going to be enough, and so you might need to have a cocktail of different medicaments that can help to block a certain type of behavior. So this would be an example in using a nucleoside analog to inhibit the enzymes of HIV-1, for instance. So here you would use, here's azithromycin, right here. What's interesting about this is that there's this azide functional group, this N3 functional group. It just replaces the hydroxyl group of deoxythymidine. That is the only difference, and that's what we call a nucleoside analog. Again, it's a nucleoside because do you see phosphate anywhere here? You don't. You have your nitrogenous base, you have your pentose sugar, you do not have your phosphate groups. That's why it's a nucleoside instead of a nucleotide. But the difference is in how this can be, and again, if we're talking about a nucleoside analog, we're anticipating that the HIV-1 virus has already entered into the cell. It's trying to replicate its RNA, right? Its mRNA and all that other stuff, not to make viral proteins, but just trying to get enough of its own coding, small segment coding, in order to eventually get there and make some DNA and or, some proteins. That is the area where we're talking about. Transcription is being interrupted at the very first part. And this would be an example of the medication that could be used to do that. And again, here's some other options. Other approaches include inhibit integrase, which we already talked about. So raltegravir, you have sosquinavir, ritonavir, osamiprenavir, and all other things, right? And even cell entry was Maraviroc and all these other, and fubarotide. All of these seek to inhibit that HIV-1 virus from either, attaching and or, then penetrating or replicating within the cell. So it's no surprise that a cocktail of this might have some benefit, okay? How else can we do it? So we just talked about a nucleoside. Well, I want to talk about a nucleotide, okay? So let's go back to the viral RNA, all right? So let's just say You're dealing with a retrovirus. Again, a retrovirus has viral RNA. It's going to infect the cell, and then it's going to make its own DNA, and proteins are going to be, created and everything else. Okay, and bad things are going to happen. So what about if there could be an analog that is created, right, that can actually short circuit or inhibit RNA polymerase? So remember how we talked about the stop codons and the start codons a little while back, where methionine was important for the start, and then there was a specific triplet sequence that stopped coding. A drug, outside the fact, right? Some of the pharmaceuticals can be made to be able to see if that can be attempted. So what about this one, where you have adenosine nucleotide analog that could interfere with the RNA chain? How does it do it? It interrupts that RNA polymerase. Basically short circuits it, because it either prematurely terminates that RNA replication process, or it mutates it so that it can't be reproduced. And if it mutates it on several different levels, then it's like, well, then you can't get your sequence going, then it'll never continue. So let's consider an example then of a nucleotide, right? Analog, a drug. How about remdesivir, which you might recognize? It's an adenosine nucleotide analog. It induces a cyanide group. Where? Right here. So you have this cyclic, pentose again, right? Where you have a couple hydroxyl groups at the very bottom, but look at that extended structure there, where you have like the cyanide group, the C and the N, right? Well, look at adenosine. Adenosine didn't have that, it just had the H popped off of there, right? And so that is where the functional substituent was done. And this is a multiple step process, although it looks like it's simple. Oh, just replace an H with the CN group and whatnot. Well, it's a little bit more challenging than that. You have to go through a bunch of different synthetic steps. However, it's not impossible, but it takes a lot of really interesting creativity. And I think Gilead Sciences pharmaceutical company did this, and they created this drug. Why did they create this drug? Why would they even do that? So I try to show you there where the analog substitutions are going. Guys, this drug was used to combat the Ebola virus, and it's currently proposed for treatment against this COVID-19 because it was effective against other coronaviruses, including the MERS and the SARS coronaviruses. So this would be an example of having a nucleotide drug that can combat a retrovirus that maybe isn't as severe, for instance, as an HIV virus. I say quote unquote severe. I mean, obviously all these viruses, if they're able to replicate and cause damage in the body, they're all going to be very horrible, right? We don't want any of that. But this would be a more about common case where, whether it's an Ebola virus or whether it's the SARS or MERS, or even if it's the COVID-19 that's just coming out, that's what's going on. So, which brings up the fact then, well, what about vaccines, right? Because some vaccines, it all depends what type of vaccine that you're having. So just to let you know, vaccines help promote your body's antibody plus T cells. So vaccines are really useful in getting your body to be really, really strong. And if you've ever had an allergy against something, you might have allergy shots, but that's just to try to build up your body's tolerance. It is especially helpful the younger that you are. The older that you are, it's way harder because, well, your somatic cells have already had so much experience. So this is where inexperience counts and matters if we're talking about vaccines. But not all vaccines can exist in the body to the same duration. And, so this is why, for instance, flu shots are generally recommended. Obviously, there's different strains of flu and stuff, and why they're even interested in this, it's kind of interesting. Like, here's the CDC. They reported in 2018 to 2019 flu season that killed 50,000 people, it hospitalized 500K, and affected nearly 40 million. Okay, so wouldn't it be nice to have a universal flu vaccine, which they don't have. They try to guess, right, on which one's going to be prevalent. This is all based on probability and obviously patterns and stuff, and definitely beyond the scope of what we're doing here, but you can look into that separately if you want. But what I did find that I wanted to share with you, this was published in Cancer Medicine and in Vaccine. Here was an interesting article that talked about the durability difference of various vaccines over time and how much protection that it gives. So here's a pertussis kind of vaccine approach. Look at how short of a period of time that it provides protection from. Virtually, first couple years, and then no protection, especially as you get it to be 25 years of age. You don't have any immunity against pertussis if you were to contract it. Diphtheria, tetanus, actually, look how long. I mean, you get some of this tetanus shots, you're good for almost your entire life. You know, more recently, I got a booster just recently, and I fall between 25 and 50, so I can be pretty sure that I'm good until my dying day. What's another one? Smallpox. Smallpox is only good for maybe 23 years, where it starts to fade out then. But measles and rubella, really good. They give you almost lifetime immunity. HPV gives you, oh, I don't know, maybe 25 years plus, all the way up to 30 years, because it's just been newer. So pretty interesting. But this is another interesting case of saying, well, geez, you know, even if you've got a vaccine, how long is it good for? And really, it's going to be obviously person dependent, of course, because everybody's going to have some different experiences with their bodies and be susceptible or be less prone, in fact, be resistant to other insults, depending on our viral makeup, our DNA makeup, right? Our genetic makeup, which genetic includes all of that, you know. But what they say about the seasonal flu vaccine, guess what? It lasts about 90 days. And it's regardless of whether it's used from live or dead viruses. And again, I just want to remind you that vaccines could contain either reduced potency, meaning that they're live, they're weakened, okay? Or, I mean, look at this. They could be a weakened pathogen with this gene stitched into a benign virus. Remember how I said not all viruses are bad? Of course not, okay? Not only is up to 8% of our genetic makeup comprised of viral fragments, but also some of the vaccines we may get could have some of its genes bioengineered, stitched into a benign virus that are then given to us, okay? So that's all part of reduced potency. They could either be dead, or again, it could be genetically modified. I guess that kind of, completely unique, where it's not even the weakened pathogen, it's just completely modified virus, okay? So you can see that we shouldn't just view viruses as this awful enemy. If we can tailor them, if we can control them and use them for really interesting purposes, then that can be super important. And what's going on right now is that there's a lot of recombinant DNA technology research that is already going on to be able to do that, because I mean, obviously we have vaccines and there's vaccine development. Anybody who wants to get into research on this, or just even be, I don't know, interested in just learning more about it, well, you can. You know, this is where you have two different sources of DNA, they're spliced together. For instance, you have like an artificial virus that you make versus, the actual pathogenic species, and then you marry them up, okay? Genetic engineering, or gene cloning as it's called, right? First step is to identify and isolate a gene of interest using a restriction enzyme. And the restriction enzyme cuts the DNA at very specific points. And then you insert other DNA into that area and use a vector, meaning it's either a plasmid, meaning it's a circular piece of DNA. And here's a cartoon that kind of shows about some of this stuff. You can look at it on your own time, but you basically have an intact plasmid, and then you have your donor part, and you excise some of that, you implant the other part, and then you replicate it. So, and then whenever proteins are produced in the manner, you say that they are expressed. And then, why is this important? Well, insulin is produced this way. This is one example. So instead of using the intestines of pigs, which just sounds crazy, I mean, how many things could be wrong with that? Whether, you're just, harvesting, intestines from pigs just for that use, I mean, how are the pigs treated? I mean, there's a whole bunch of different things that can go off of that. But if you could do this in a recombinant way using DNA technology, that's pretty cool, and it might be even safer. Could even potentially be, less costly. This is also present in genetic testing, fluorescent labeling of reporter genes, and genetically modified crops. All of this uses DNA technology. This is part of our life, and this is what's going on. It's only going to, continue to be used. And CRISPR is really this type of genetic modification that is just another step in the evolution of recombinant DNA technology. Okay, and there's lots of different ways to extract DNA. Okay, this is, you have all these different parts where you have DNA extracted from a blood sample, you have it broken into many of the base pairs. Okay, so then it's sliced up, and then they're, then they're combined with other types of bacterial DNA, and you do that for replication purposes. Okay, and you can do all this in order to be able to study something. Okay, so all of this shares the common features of it. For me, I have a demo that I want you guys to take a look at. It is on YouTube. There's 4 videos. They're all short, and please read the description because I think it's helpful. What you're looking at here in this beaker is actually banana DNA, and I tried to talk about it a little bit. You know, I wanted to use something quite big. You know, bananas are obviously a big fruit. You know, you could use other things. I mean, you could do a cheek swab, right? You could use other fruits. You could, you know, whatever it is. But I thought this was kind of interesting. It's an easy experiment that you could do just to show that you could get it. But what's pretty interesting about it is whenever I filtered it, again, you didn't see any of this DNA at first whenever I had the filtered material. It existed as a filtrate, and then it only fell out of solution by virtue of the addition of a pretty high-grade ethanol product. I just used denatured ethanol, which is about 86%, and that in of itself disrupted hydrogen bonding enough with the environment, right? The DNA from the solvation, you know, from the water in which it was collected. Because I started with water, by the way, and, you know, I mashed it up just gently. You don't want to do it too much. You don't want to damage the DNA. But then using ethanol, you can selectively excise that extract it, precipitate it, whatever you want to call it, from the solution, and then you can handle it. So in the course of handling it, you could break it and other things. And obviously, if it sits for a long amount of time, the DNA will break up because there are going to be limitations, and the ethanol will continue to interrupt those hydrogen bonding forces. But it can be done. And, you know, if you don't want to do it the way that I show you in the YouTube, here's another way that you can do this. And this is using onion. One of the interesting cases of using onion, by the way, is using this Proteus meat tenderizer, which tries to help break down some of that tough cellular walls of the onion, you know, plant in and of itself. This may seem a lot more complicated. This is just one example. This is shown in the American Museum of Natural History. If you take a look at what I did, I think you might like it. But, you know, and with that, that's it. So that is the end of the entire chapter of 11. And next thing you guys got to do is work on the homework and exam. We'll go from there. But check out that YouTube video, and the lab, the corresponding simulation lab, ought to help as well. Okay. So there you have it, RNA and DNA.